And without delay, I would like to invite our second presenter, who is Ghadeer from Syria, and the title of presentations is Anti-Defense Systems of as Maltophilia. So over to you, Ghadeer.
Good evening, everyone. Can you hear me?
Yeah, we can see and hear as well, so you're good to go.
Okay, excellent. I would like to thank the organizers for setting up this webinar and the speakers. My name is Ghadeer Jdeed. I did my PhD research at the Institute of Chemical Biology and Fundamental Medicine, Russian Academy of Science. And my talk today would be about the defense repertoire of Stenotrophomus maltophilia. So when we study usually phage therapy, we focus on the properties of phages, like we study if they are lytic, if they are specific against the target strain of the patient, and then we see if they are usable or not. But the other side of the story is the bacteria itself, because over the evolutionary history of bacteria and phages, they developed systems, mechanisms, and methods to defend themselves against the phages. So here I studied how Stenotrophomus maltophilia actually developed ways against phages. So what are these bacteria first? Stenotrophomus maltophilia are gram-negative bacteria. They can form biofilms on medical devices. They are considered by World Health Organization as emerging pathogens, and they are highly intrinsic. They have high intrinsic resistance to antibiotics. In this picture, we have 5 strains that we locally isolated and sequenced, and we see the huge number of operons responsible for antibiotic resistance in them. So for this research, I did pangenome analysis of locally isolated and also global Stenotrophomus maltophilia strains that were downloaded from GenBank database. And then I identified antiphage defense systems in them, and also determined defense islands. And I will talk about these later. First, the pangenome analysis showed that these bacteria that we studied 72 strains, are highly heterogenic, meaning they share a low percentage of genes among them. We see here this block of genes that are shared between the strains. These are called core genes. They are shared among all the strains, and they constitute only 4% of the genes. That's it. And this is actually quite known about Stenotrophomus maltophilia, that they vary among themselves, and they constitute what's called Stenotrophomus maltophilia complex. There are some suggestions even that they should be divided into new species. Okay. So after that, I looked into the defense systems that these strains have, and it turns out that there are 500 instances of defense systems that belong to 72 types. These types can be divided by their function to phage protein sensing systems, abortive infection, restriction modification, and restriction modification-like systems, and sequence-independent detection of phage nucleic acids, toxin antitoxin systems, and also many systems with unknown functions. The most widely spread systems were RIC-BCD and WADGET1. They were present in most of the study strains. They were followed also by restriction modification systems, different types of them, like 1, 2, 2G, 3, 4, which were present in around 20 to 40% of the systems. We also had systems GABIJA and CIP2, which were present in around 50% of the strains that we studied. So other systems were very rare, like I would find them in 1 or 2 of the studied strains. But these systems, restriction modification systems, RIC-BCD, and 1 abortive infection system were found in around 50% of the strains. Then I looked at the variability of these systems, meaning how different they are among strains. And the darker the colors we see here, the more conserved these systems are among the strains. It turns out that most of the systems were highly variable among the strains. For example, restriction modification systems, type 1, 2, 3, 2G, and 4, were conserved at around 30% only among the strains, meaning that in most cases, each system differed from the system found in other strains by around 70% of its amino acids. Mostly it's due to adaptability to local phages, apparently. Some systems were conserved, like system BRICS1, abortive infection system ABA4, and several genes of WAGET1 and also RIC-BCD were highly conserved. But in most cases, defense systems were highly variable among strains. Then we looked at the conserved genomic fragments of the bacteria where these systems could be found. And it turns out that there are specific genomic regions, we call them, based on previous research, defense islands, where these systems aggregate. We found 27 defense islands, and 4 of these defense islands contained 60% of all the systems that we found. So even though the strains were very heterogeneous, even though the systems were very different, it turns out that in 4 locations of the bacteria, 60% of the systems were found. Here I present 2 examples of these defense islands, defense island 27 and 4. Defense island 27 contained all instances of RIC-BCD and most instances of WAGET1. And what distinguishes defense islands is that they are flanked by core genes. Again, core genes represent only 4% of all the genes in Syntrophorus multifilium complex. We see them here in blue. So again, defense island 27, it contained 25% of all instances of defense systems. And then there's defense island 4, which contained 16% of all defense system instances. It contained most of restriction modification type 2, in addition to other systems. Then we looked if there's a relation between how related a strain is to another strain and the chances of containing identical systems in identical defense islands. And it turns out that in cases of phylogenetically related strains, even when they are isolated from different locations or different countries, they would contain the same defense islands. So in this example, defense island 19, we see 5 strains, and 3 of them are phylogenetically related. These 3 contain the same defense system called Sophis. The 4th one, which is distant from them, contained another system, Ghadeer, but the 3 that were related contained identical systems. Another example is here. We see 2 strains that are phylogenetically related, and they contain the same 2 systems, BRICS1 and WAGET1. But we also found, surprisingly, that in many cases, strains that are unrelated phylogenetically, that are isolated from different locations, would still contain the same defense systems. Examples are here. These 3 strains are distant from each other, and yet they contain DSR1 system. And these 4 strains also contain...
Sorry, could you please conclude soon?
Yeah, and these 4 strains contained SDUA system. And again, we found many systems of defense in these bacteria. Interestingly, 4 defense islands contain 60% of the systems. And actually, this allows us in the future to find more systems because, again, they tend to aggregate in the same locations. You can check out the paper that we published, and thank you.